Hello students, welcome to Al Noor International School Virtual Learning. In this session, you will learn Chapter 15, Lesson 3, Applications of Genetic Engineering. Students, you can find this lesson on your textbook page 431 to 434. Let's see the lesson objectives. The first objective is to describe the benefits of genetic engineering as they relate to agriculture and industry. Secondly, explain how recombinant DNA technology can improve human health. And the third one is to summarize the process of DNA fingerprinting and explain its uses. Well, let's get started with the application of genetic engineering in agriculture and industry. So genetic engineers work to improve the products we get from plants and animals. So genetically modified crops may be more nutritious or higher yielding. They may be resistant to insects, diseases, or spoilage. Some can produce plastics. Genetically modified animals may produce more milk, have leaner meat, or contain higher levels of nutritious compounds. Transgenic salmon grow rapidly in captivity. Transgenic goats produce spider silk in their milk. Now let's see some of the application of genetic engineering in health and medicine. Recombinant DNA studies are leading to advance in the prevention and treatment of disease. Examples include vitamin-rich rice, human proteins made in animals, animal models of human disease for research, and bacteria that produce human insulin. Gene therapy is a process of changing a gene to treat a disorder. However, gene therapy is still an experimental and high-risk technique. And genetic testing can identify hundreds of inherited disorders. Not all genes are active in every cell. Now let's see what microarray is. Microarray technology lets scientists study thousands of genes at once to determine their activity level. And the other application of genetic engineering is in personal identification. What do you mean by DNA fingerprinting? DNA fingerprinting analyzes sections of DNA that may have little or no function, but that vary from one individual to another. DNA fingerprinting is used in forensic, the scientific study of crime scene evidence, to identify criminals. It's also used to identify the biological father when paternity is in question. Common ancestry can sometimes be determined using mitochondrial DNA and Y chromosome analysis. Students, the above explanation is simply the general overview of the lesson. For more information, for detailed information, please watch the links below. And after watching these videos, you can proceed to the section assessment part. You can find the section assessment part on your textbook page 434. Assessment part, section assessment 15.3. Okay, question number 1A. Give 2 practical applications for transgenic plants and 2 for transgenic animals. I hope you are trying the answer. Well then, so sample answers could be plants, higher yields, resistance to herbicides, and animals, increased milk production and leaner meat. Let's try another one. 1B. What might happen if genetically modified fish were introduced into an aquaculture facility? Here is the sample answer for you. If genetically modified fish were introduced into an aquaculture facility, the fish populations might crossbreed and produce offspring with new characteristics. Let's go to question 2A. Name 3 uses for recombinant DNA technology. Well then, so it could be preventing disease, medical research, and treating disease. 3A. List the steps in DNA fingerprinting. Well, the steps are: restriction enzymes cut a small sample of human DNA, gel electrophoresis separates the restriction fragments by size, a DNA probe detects the fragments that have highly variable regions, revealing a series of variously sized DNA bonds that can be matched to another sample. Let's go to B. Medicines in the body interact with the body's proteins. How might normal variations in your genes affect your response to different medicines? Well then, so variations in genes result in variation in proteins. Because genes direct the building of proteins, as a result, the interaction of medicines with proteins varies from person to person. Let's see another one. Why is DNA fingerprinting more accurate if the samples are cut with more than one restriction enzymes? Yes, each restriction enzyme cuts DNA at a specific sequence of nucleotides. The more restriction enzymes that are used, the more DNA fragments will be produced. The more fragments there are to separate and analyze, the easier it will be match the resulting DNA fingerprint to another